Where do scientists start from when seeking new pharmaceutical products? Even with today's modern techniques of computer-aided design and mass synthesis of candidate molecules by combinatorial chemistry, the answer is often in natural products or metabolites found in living organisms. The drug aspirin is based on a molecule derived from willow bark, which is an ingredient in several traditional remedies for fever. The molecule responsible for this action was extracted and identified by 19th century chemists who made a small change to its structure to increase its efficacy. Screening for promising candidate molecules may be a long and time-consuming process. The anti-cancer drug Taxol was first identified as a candidate for research by extracting and purifying natural products from tens of thousands of plant species, including the Pacific yew, from which Taxol was first isolated. Modern screening techniques seek to establish pharmacokinetic features of the candidate molecules. For example, the strength of bonding between the molecule and receptor sites, including enzyme active sites. This permits in vitro screening, in other words, using components of an organism such as enzymes that have been isolated from their usual surroundings. Tests can be automated and carried out very rapidly on a large number of candidate molecules simultaneously. Screening is often carried out using computer modeling to predict the likely effect of a molecule on a receptor site, a process often referred to as in silico screening. If in vitro or in silico screening gives a promising result, the effect of the candidate molecule on living cells must be established. Bacteria will be exposed to potential antibiotics, or the growth of cancer cells in the presence of anti-cancer agents will be assessed. The next stage will be to carry out animal studies to assess the effect of the drug on a living organism. Not only will this give valuable information on the efficacy of the molecule in treating the disease, but it also allows a range of data about toxicity to be obtained. For example, this will include the LD50 value of the drug, the dose required to cause death in 50% of individuals of a particular species. The data from animal trials will then be used to calculate suitable dosage levels to be used in human clinical trials. Several phases of trials are necessary. First of all, to establish the safety of the drug, and then to gain reliable data about the efficacy of the drug. Trials must then be set up and run very carefully to ensure ethical integrity and reliability of evidence. Although Phase 1 trials to establish a safe dose involve healthy volunteers, Phases 2 and 3 require sufferers of the disease to be tested. If Phase 2 proves promising, then large-scale Phase 3 trials will be carried out, comparing the response of one group of patients given the drug with that of a similar group of patients taking a placebo drug, or the best treatment currently available. Explaining the implications of taking part in a trial with an unlicensed drug and obtaining informed consent from the patient is a delicate and important job. When all the data from animal testing and clinical trials has been obtained, an application is made to the relevant body for a license to manufacture and market the drug. The whole development process from initial screening to the product being available in pharmacies may take anything up to 15 years. Of the tens of thousands of molecules that may have shown promise at the screening stage, only one will have made it through to licensing. The costs can be staggering, and £150 million to bring a product to market is a typical figure.